Good morning, everyone. And at the outset, I'm really grateful to all the organizers to give me this opportunity to share some of our work. So we work on human pathogens. Tuberculosis is one of them, HIV and SARS-CoV-2. And our laboratory is in Indian Institute of Science, Bangalore, which is not too far from here, 20 minutes, 25 minutes drive. Most of the work is actually done at Center for Infectious Disease Research. And as you can imagine, that M. tuberculosis is a BSL-3 class pathogen, so we really need a state-of-the-art BSL-3 facility, which we have at Indian Institute of Science, which is one of the biggest BSL-3 facility in India. And we have a facility to do TB experiment, also to do any bacterial pathogens which require BSL-3. We have the facility. We also have facility to do viral infections and viruses which require BSL-3. And also, we are making a new animal BSL-3. And these BSL-3s are actually open for everyone, from academic setting to private settings within India and outside, so you can have access. And we have actually quite a lot of joint projects funded by BBSRC, MRC, or NIH within this facility. So with this, the only point I want to add is that ISC is also constructing a 800-bed medical hospital, which is going to open probably next year. And then we will have access to quite a lot of clinical samples, and there'll be a clinical trial center within ISC. So this is going to be a really nice combination of fundamental and medical science coming together in a given institute. So with that, I'm going to talk about tuberculosis and our effort to understand phenotypic AMR. As the word suggests, phenotypic, therefore, this is a physiological, drug tolerance which pathogen experiences or induces without inducing any genetic changes. So it's not a heritable changes which we generally talk when we discuss drug-resistant infections. So in case of tuberculosis, the major problem is long therapy time. If you look at tuberculosis treatment, it is about 6 months. The most non-complicated form of TB requires 6 months of treatment, where 2 months of intensive phase, where 4 drugs are given, and then 4 months of continuation phase, 2 drugs are given. And this is a picture of bacilli in the sputum of patient who is on therapy. And you can look at that early on, within 2 months, there is the kinetics of killing of bacteria is actually quite rapid, followed by delayed kinetics of killing. And this kind of phenotypic heterogeneity is actually the basis of long therapy time required to treat this infection. And we have some idea that there are actually 2 subpopulation of MTB in your lungs. One is which is actively replicating and quite susceptible to anti-TB drug, and perhaps the other one metabolically quiescent, non-replicating, slowly dividing bacteria, which takes quite a long to get cleared. And our hope is that if we understand this process, we can come up with a combination therapy that can convert this curve into a log linear curve, and that's where we wanted to understand this process. And if you look at a tuberculosis infection, tuberculosis lesion, 70% of infection happens in lungs. And you can look at tuberculosis lesions, and these lesions contain granuloma-like structure, which is a collection of immune cells, lymphocyte, macrophages, neutrophils at the cuff, fibroblast. And this kind of structure, MTB, is actually residing. And this also exposes MTB to certain immune pressures that allowed it to adapt in this environment. Also, there is loss of vasculature, so the drugs are actually not going to permeate in this environment as you would like to. And then because of these immune pressures, which MTB senses and phenotypically switched into a state where it is transiently tolerant to drugs. And this transient tolerance is very important because it provides phenotypic AMR. It gives MTB to actually stay live in response to anti-TB drugs. And then that time is actually utilized to convert into a MDR or XDR form of tuberculosis. So if we really have to target MDR or XDR TB, we have to look at the step back, and we have to look at phenotypic AMR, which sort of potentiate or mobilize mechanism to convert this bacteria into an MDR and XDR tuberculosis. So we're really looking into a phenotypic AMR, and we wanted to understand what all host immune pressures are sensed by this bacteria to switch into a phenotypic AMR state. So we did an experiment where we have taken naive bone marrow-derived macrophages infected with mycobacterium tuberculosis and then exposed to anti-TB drug moxifloxacin at a 3x MIC, and you see that after 96 hours of treatment, about 20% of bacteria is surviving this treatment. However, very interestingly, if we activate these macrophages by interferon gamma, that sort of induces a lot of stresses on bacteria. This proportion of drug-tolerant bacteria increased to about 50%. So what it is telling you that some of these signals which are induced by interferon gamma, like nitric oxide, reactive oxygen species, low pH, which is how this environment of macrophages are going to look, they are being sensed by these bacteria to become drug tolerant. And one of the example is that if you can take away ability of macrophages to produce nitric oxide by using interferon NOS to knock out mice, and then you activate by interferon gamma, you will see that this level comes back to 21%. So it's not that you're getting absolute eradication of bacteria, but delta is important here. You do see quite a lot of decrease in drug tolerance if you take away nitric oxide producing ability of macrophages. So this is very important for us because nitric oxide can affect bacterial physiology in many different ways, and one of the ways is that it can affect bacterial redox state. Bacterial ability to resist oxidative stress or bacterial ability to undergo redox changes is very important, looks like, because of this data, and therefore it's possible that some of these alterations could lead to phenotypic AMR. So that is why we wanted to ask the question, what is the redox state of bacteria when it is inside macrophages? And can we answer this question by measuring the redox state of bacteria inside macrophages? And for that, a couple of years back, about a decade back, we developed this biosensor, which is a genetically encoded fluorescent biosensor, ratiometric biosensor that you can express in MTB, for that matter in any bacteria. And you can measure the level of oxidative stress bacteria is experiencing when it is growing in vitro and when it is inside macrophages. So I'm not going to go into details of it, but what we found that upon infection, these bacteria, when they enter into macrophages, they appear to be in a different redox state. Some of these macrophages contain bacteria in an oxidized redox state, which means they are under oxidative stress. Some of these macrophages does not alter the redox state of bacteria, which we call it basal. And some of these bacteria seems to be quite reduced, which means that they have high antioxidant capacity or they have high ability to deal with oxidative stress. We further did a lot of experiment to connect it to phenotypic AMR, and we found that those bacteria which are under oxidative stress, they are killed faster by anti-TB drugs, followed by basal, and the bacteria which have a high antioxidant capacity, they are quite tolerant to anti-TB drugs, quite robust. And it seems that at least these bacteria are responsible to some extent for long therapy treatment time. And this is what our focus of our lab is, to understand the physiology of these bacteria and if they are responsible for the prolonged treatment of TB. So what we did to understand the mechanism, we sort of sorted out these macrophages, and we actually taken out bacteria from these macrophages and then isolated RNA of bacteria. So it's a very complicated experiment done in BSL-3. We have a FACS sorter inside BSL-3. So you can actually sort out infected macrophages, take out bacteria, take out the RNA, perform the RNA-seq. And I'm not going to go into details. There are several mechanisms. One of the mechanisms which is playing out in drug-tolerant bacteria is their ability to actually dispose cysteine. A lot of mechanisms that utilizes cysteine to different processes like iron sulfur cluster biogenesis, hydrogen sulfide gas, and production of antioxidant mycothiol are quite induced in drug-tolerant reduced bacteria. And this seems to be interesting because if there is an accumulation of cysteine, cysteine accumulation can drive Fenton reaction to produce more ROS, and bacteria could die. So one of the mechanisms by which MTB can protect itself from dying inside macrophages is to take care of excess cysteine. And that gave us a clue that if cysteine metabolism is so important, can we overload bacteria with cysteine and kill them by anti-TB drugs? So we did a very simple experiment using this health supplement N-acetylcysteine, which is readily available, non-toxic, used to reduce drug toxicity. And also it is a precursor of antioxidant like glutathione. So we did this experiment in mice where we supplemented N-acetylcysteine with the anti-TB drug moxifloxacin. After 21 days of infection, we treated mice for about 10 days. So it's like a pilot scale experiment. And you would see that this is a CFU in the lung of infected mice. After treatment, you see decline in CFU, but the decline is much more in case of moxi plus N-acetylcysteine. However, the more important experiment is that by addition of N-acetylcysteine, we were able to suppress the emergence of drug resistance. And this experiment is also done in mice where we have done the similar kind of experiment, but then we scored for moxifloxacin resistant bacteria under different treatment regimen. And here we found that moxifloxacin alone increases resistance probability by 67%. However, if you combine moxi plus NEC, that comes back to 17%, which is similar to untreated mice. So just by simple addition of N-acetylcysteine, we were able to overload bacteria with cysteine. We are able to reverse reductive stress or reductive redox balance, and we are able to make bacteria more sensitive to anti-TB drugs. So we moved on, and this time we did RNA-seq of host. So rather than isolating bacteria, we sort of sorted out macrophages, isolated RNA of macrophages, and then asked the question, how are these macrophages different? Because different macrophages are harboring bacteria in a different redox state and different state of antibiotic sensitivity. So we did RNA-seq of macrophages, and one thing which is very clearly stand out that macrophages which are containing drug-tolerant bacteria, they are inducing TCA cycle. So mitochondrial metabolism is up, mitochondrial respiration is up, TCA cycle is up. So we did a lot of phenotypic experiment on these macrophages after sorting, and we came up with a list of changes which these macrophages are ongoing. So macrophages which are containing drug-tolerant bacteria, they are showing high oxidative phosphorylation, which means high mitochondrial activity. They are low on glycolysis. They also produce less mitochondrial ROS, and these macrophages are showing acidic pH. However, those macrophages which are containing antibiotic susceptible bacteria, they have all these parameters in a reverse direction. So based on this fundamental observation, we thought that there is a potential for host-directed therapy wherein we can switch some of these parameters. For example, you can switch acidic pH to neutral pH by using anti-malarial drug chloroquine, which is already in use, very safe drug. Also, you can induce glycolysis using this FDA-approved drug maclagine, which is an over-the-counter drug used to treat nausea and vomiting. So we did an animal experiment, to show that by adding chloroquine with the frontline anti-TB drug isoniazid, we are able to sterilize the lungs of mice infected with TB much faster than isoniazid alone. At this point, we are unable to get any bacteria out of lungs. So there was total sterilization of bacteria in the lungs. Similarly, we use maclagine, and here you can see that maclagine it is able to induce more oxidative stress in bacteria, and when you are combining maclagine with isoniazid, look at the profile of isoniazid alone, where it is decline the growth, but maclagine co-treatment with maclagine is further potentiating the effect. So this is a pilot scale experiment. We are going for long-term experiment, and these are some of the examples wherein fundamental science can drive us to look into very sort of easily affordable solution to look into repurposable drugs like chloroquine, like maclagine, like N-acetylcysteine. So these are some of the examples, and we are filing patents, and we are also trying to get clinical trial going for some of these combinations. So with this, I'll end. This is my group, and I'll gratefully acknowledge all the funding agencies who have funded our science for a decade now, and all the collaborators at NCBS and ISC. And I'll be happy to take any question. Thanks. Right. So that's again very interesting question, because these could be also biomarkers. Yeah. So we have to think whether generally biomarker based on host profile is based on transcriptomics, and there are some studies where looks like neutrophil signature is predominant. So we have to see whether these RNA signature which we are finding is actually also happening within the patients. What we showed is very focused macrophages transcriptome. So, you have to look into whether macrophages are giving that profile, or if you look into blood. Yeah.
So one more.
Right, so we have done those experiments and already published. So if we expose TB containing biosensor in vitro in a synthetic media to anti-TB drugs, none of these anti-TB drugs induce oxidative stress. But if you repeat the same experiment when bacteria is inside macrophages, all of them induce oxidative stress. So it's the mode of action and the way these antibiotics are going to kill MTB inside host is going to be very different than in vitro and synthetic media. It has an implication for drug screening that most of the drug screening, if you're doing in 96 well plate in synthetic media, the chances that you're going to hit a new drug which is going to kill bacteria effectively inside macrophages or host is going to be minimum. So probably drug screening has to move to macrophages or infected macrophages.
So in the interest of time, we'll take the other questions during tea time.
Sure.
Yeah? Thank you.
Thanks.